Okay, do you see the recording? Okay, it has started, right? All right. Well, so it is my pleasure to introduce Davide Micheletto, fellow of the Royal Society from the University of Edinburgh, who will talk to us about new methods combining AI, knot theory, and biology. And thank you very much for making the time to give a talk this late time there. So thank you.
Thank you, Danny. So I realized that my abstract was very focused on machine learning and knots, but actually, I then realized that the audience might be more interested also on the more bio stuff that we do. So I've included a couple of sections in the talk where I'm going to talk about more biological stuff before I get into the machine learning knots and topological invariants. So. My name is Davide Micheletto. I'm a group leader at the University of Edinburgh, and today I'm gonna give you a little bit of view of what we're doing. Before telling you about the story that I've got more or less 3 stories I want to tell you about, but I'm gonna tell you about I'm gonna tell you about something that I cannot tell you about in this talk. So kind of give you a little bit of an overview of what my group does. So we do a lot of material science and complex fluids, the type of stuff. So we look at viscoelasticity of DNA, for instance, of DNA dense solutions, for instance, as a function of supercoiling. We make DNA gels. So this at the top right is a hydrogel purely made of DNA and water. So we make it in the lab, but also we do simulations to understand its serological properties. So we perform bulk rheology to measure its viscoelastic properties, and we couple simulations and experiments to understand its properties as a function of the topology, let's say, of the DNA that we can building blocks. We do a bit of genome organization as well. So this is more kind of work that during my postdoc, but then I still there a little bit. So this is an example of simulation where I simulated a little piece of chromatin, so DNA in a coarse grain model. Our DNA wraps around histones, and to make this little, let's say, chromatin fiber, and then there is like a ring representing HIV DNA that tries to integrate into, and the question is, where does it integrate? And we find that there is a bias depending on the chromatin structure, for instance. This is 2019. This is another paper in which we do a model of polymer recoloring. So the beads, let's say, that they're colored differently, but they can also infect each other. So the blue can infect red and red can infect blue, and this is to model epigenetic marks spreading along chromatin or along the genome. And we find that with certain models you can essentially recapitulate this domain structure that people now see in how the genome is organized inside our cells. This goes back to my PhD even, but we're still doing a little bit of this. Is topological glasses. So we take polymers and we study dense solution of ring polymers or tadpole polymers or polymers with more complex architecture. And depending on when at dense solutions, there are some emergent sort of interactions or entanglements that are very different from what linear polymers have. And in some conditions, in some regimes, you can get this emergence of a glassy state that is due to the topology of the polymers rather than the temperature. So that is also quite interesting topic. And this is very recent. We started to play around with drug discovery. So there is a student that plays around with diffusion models to try to come up with new drugs for pockets and for protein pockets. And in particular, we're interested in topoisomerase, which is this protein that changes DNA topology. So these are the things that I'm not going to tell you about. And the things I'm going to tell you about today, so one is about a little bit about topoisomerase and condensin. This first part of my talk really originates from the question of how does nature organize DNA inside our cells. So this is a reconstruction of the genome inside a HeLa cell, and essentially, so perhaps you know, but for those of you who don't, each human cell has 2 meters of DNA in each cell, which is, if you scale it up, it's 20 kilometers of rope in 10 centimeters inside your cell phone. This is clearly leads to a lot of entanglements, but and even more strikingly, if you take all the DNA that is within your body, so in all your cells, you can reach to the end of the solar system and back many times. So this is quite interesting. If you think about, in each cell there is this huge mass of DNA that needs to be compacted in a small space, and obviously leads to a lot of entanglements. But still the cell can replicate. You see here cell division happens quite seamlessly and many times you're in your body. And if entanglements were a problem, you will get a lot of DNA breaks and the cells will die. So there is a way in which nature has overcome this, and we want to essentially understand what it is and perhaps also learn from it to make new materials. This is why we're also interested in materials that change topology. And so we know now then, so beyond this, I mean, if you still don't believe me, these are just some pictures that, so this is a DNA knot created on a plasmid. So on a plasmid, this is circular DNA. And this is the DNA inside a bacterium that has been kind of exploded out. So you can tell that there is a lot of entanglements, and even in chromatin. So this is an experiment where people essentially, this is a gel electrophoresis experiment where you run your DNA through a gel, and you can separate the DNA based on its topology. And in this particular experiment, the authors got some very long plasmids that introduced in yeast cell. These plasmids get chromatinized, so essentially it's chromatin. And then you run it through the gel, and you see that you form knots in these plasmids inside yeast cells. So chromatin is knotted inside our cells. Okay. So how does nature manage entanglements? So there are two very important proteins, where the first one is this topoisomerase that I mentioned before. So topoisomerase has a very interesting structure, has two gates. So I'm going to tell you about type A topoisomerase. There are other types, but I'm going to focus on type A, 2A topoisomerase. So it has two gates. So there is a top gate, then kind of another binding site here, and then another gate. So what happens is that the topoisomerase first binds one strand of DNA, and then has the top gate open for a second strand. Then when the second strand is kind of captured, ATP binds the top gate, and this second strand gets released through the bottom gate. And essentially, if you imagine two strands of DNA that are entangled, then this topoisomerase acts as a little scissor and a little glue. So it finds an entanglement, it cuts one of the two strands, of the two double strands, passes one through the other, and then seals it back. And by doing this, you change a crossing, but by changing one local crossing, you can also change the topology. In fact, if you take two rings and you do this operation, what you get is two unlinked rings. And you can also do the other way around with topoisomerase. And in some conditions, topoisomerase does this. So it does from two unlinked plasmids, let's say, it links them back. The other interesting protein that we are working on right now is called the SMC, structural maintenance of chromosomes. Condensin is also called condensin or coisin. There are different types. And so this has also a very interesting structure, and what it does, it also binds DNA at multiple places, and then it essentially cycles through conformations, and it forms this loop. Okay, and this loop it gets extruded. And what I mean with extruded is in this movie. So this is a coarse-grained kind of representation of what happens. Coisin, which is this loop extruder or this SMC, binds DNA. It's here represented by a ring, and then it kind of pulls by changing conformation. Although in this movie you don't see changing conformation, but in reality it does. It kind of pulls a loop through. Okay. And this is what people now call a loop extruder, and people and the community of genome organization think that this operation is very important, both for the mitotic chromosome structure, but also interface. So to really organize the genome inside our cells. Now, what I wanted to know is what is the evidence that these two proteins are important for genome topology? So I show you a couple of evidence. One is that if you take some cells and you inactivate the topoisomerase, so this are some cells, some mitotic chromosomes. So this is the typical image of a mitotic chromosome that you find in textbooks. Now, if you acutely remove topoisomerase, what you get is malformed chromosomes that remain all entangled. So in this case, you don't have topoisomerase, you have condensin, and the chromosomes remain entangled, which makes sense. But there is other experiments that show that even in presence of topoisomerase, so in this case you have Drosophila cells that are being through mitosis, so they are being during cell division. So in this top row here, you have the red fluffy stuff is DNA, and during you have the two cells that successfully divide. But if you don't have condensin, the two cells remain entangled through their DNA. So in this case, in these experiments, these are with Drosophila cells, you have topoisomerase, but you have an acute inactivation of condensin, and the chromosomes are still entangled. So you need both to organize this topology of the gene of the chromosomes. And to remove entanglements correctly. So we wanted to understand exactly the mechanism behind all this. And one thing that came to my mind some years ago was that was to try to understand better what loop extrusion does on a knotted polymer, for instance. So let's take, for example, a polymer, a curve that is knotted, and it's delocalized, so the knot is a bit everywhere. You cannot really tell. I mean, the curve is knotted throughout. Now imagine that I start to do this loop extrusion and I wait until it's finished. What will happen is that the knot is all localized on a short segment of the whole curve. This is obviously a sketch. I can show you a simulation where at the beginning of this movie, essentially you have the white part is the stuff that is already extruded. So I'm showing you just second half of the movie. You cannot really tell that the polymer is knotted. But if I let the movie go long enough, you start to see that the knot becomes smaller and smaller, and all localized on the dark part, like on the dark segment. And at the end, it becomes very, very small. Okay, so now you can tell that it's all there. Okay, and these are just snapshots from the simulation. So this mechanism is I always think about this mechanism as sort of a climbing rope analogy. I'm not sure if anyone climbs there in the audience, but essentially before you start to climb, you want to make sure that your rope does not contain knots, because otherwise it gets trapped on the belay. So what typically I do before going to climb, I get my messy rope, which I don't know where, if there might be a knot in there, and very likely there is, and I pass it through a carabiner. By passing it through a carabiner, you localize the knot, and eventually you can undo it more easily than in this other case before passing it through the carabiner, because you cannot really see where it is. Okay, and this is similar in some senses. So once that the knot is localized, in some sense you can imagine that it's easier for topoisomerase to identify where the knot is and to undo it. Because so this is another simulation I'm going to show you, and here, instead of having just loop extrusion, you also have the effect of a topoisomerase. So topoisomerase here is represented as some beads that are phantom. So these beads allow essentially other beads to cross through. It's not a perfect model for topoisomerase, but it's, for now, I mean, it's what most people use as a model for topoisomerase. You allow essentially the polymer to cross through itself. So again, at the start of this movie, I'm showing you just this. I'm showing you just the second half of this movie. So here there is already some loop extrusion that has occurred. So it's all the white part. The knot is already a little bit localized here, and then it gets even more localized, and then eventually, because of those green beads and because there is it's entropically favorable for the knot now to open up and to explore more configurations, right? And so now you've undone the knot. Okay, so it's quite interesting now. So that is an example in an ideal system. Now inside our cells, DNA is all packed, and so what I wanted to show is that,
David,
yeah.
what happened at the knot? How did it open up? What's happened there?
So what happens is that you have
Is there an actual knot there?
yeah. So here, you
Okay.
see, it's a trefoil knot.
And somehow it crosses through itself to
The
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 end
black,
then?
yeah, the black beads can cross through the green.
The green, so
So
the topoisomerase,
the green is this phantom
yes,
bead that
right.
allow anything to pass through.
Yes, which
So it
is
kind of
kind of modeling the action of the what it does to DNA, right?
topoisomerase, exactly. It
Okay,
allows the
okay,
crossing, and now it's open.
okay,
Okay.
thank you.
So now I want to show also in a more confined situation. So now I have a simulation in which the DNA, the polymer is all confined, and it's a very complicated knot. I think it's a 7-1 in this case. And I'm not gonna go through the whole movie, but essentially there is a very complicated knot all within a sphere, okay? And little by little, essentially this knot gets simplified down to the unknot through going from 7-1, 5-1, 3-1, and a knot. And I guess here the important thing is a comparison, right? So what we did was to compare this synergistic effect of loop extrusion and topoisomerase, which is this synergistic here tables, sort of columns, against RP is the random passage. So random passage is when you have topoisomerase but there is no loop extrusion. Okay, so if you let's compare these two. So let's say that have the passage, so the knot transition between the 7-1 and the 5-1. If I have just random passage confined, so this happens 2% of the cases, or 30% of the cases if it is completely free, it's open, right? Instead of being confined. So it's especially confined is very unlikely that the 7-1 becomes a 5-1 in this case. But if I have this synergistic mechanism, so I have loop extrusion plus topoisomerase, you have essentially 98% or 92% of the transitions are towards simplification. And so you really kind of introduce this. So we've done it on many different and complicated knots, and by having this synergy between loop extrusion and topoisomerase is the way to kind of systematically simplify even complex topologies under confinement towards the unknot. Because otherwise, if you just had just topoisomerase, let's say phantom beads, it's natural to think that if under confinement you create more complex topologies. It's like your headphones inside your pockets, right? You get knots eventually. Okay, so this was I think one part of the story, and oh yeah, there is a little subtlety here which is quite interesting, and perhaps is interesting for more people interested in topology. So when I was doing this, then I asked myself, but is it really due to knot localization? Because inside our cells, you can think that it's very difficult to completely localize a knot because there are proteins, there are other things bound to DNA, there is chromatin. I mean, it's much more complicated than that. So I could believe this mechanism for naked DNA, but for chromatin is much more difficult to imagine. So what I did here was to do a simulation where I have multiple loop extruders. So I have on the left hand side here you will see 2 loop extruders. On this is polymer is knotted. And then on the right hand side, yeah, the blue part, the cyan part, is the shortest arc that you can define knotted by essentially cutting it and joining the ends. Is the shortest arc that you can define knotted. So let me skip a little bit ahead. So if you have 2 loop extruders, there is a competition between the two, and actually you don't confine the knot. So the knot here becomes it stays very delocalized, and it stays delocalized because I'll show you in this. This is a chymograph essentially. These are on the y-axis. You see these are the feet of the loop extruders that start from a little segment and then they open up as they walk in different directions. And the gray part here is this region that you can define knotted. And if you have one inside and one outside this knotted region, what happens is that what you do, you don't localize the knot, but you localize the essential entanglements. So what we've done here essentially is what the loop extruders have done is to localize the essential entanglements, the essential crossings of the knot. So rather than the whole knot itself. And also in this case, eventually you get unknotted, systematically unknotted. So it's not quite only the knot localization, but it's about localizing the essential crossings. So the conclusions for this part is that the synergy of condensin and topoisomerase is necessary for systematic unknotting, or at least in this model. But we've done experiments, and there is another side to this story, in the sense that the experiments we have done in our lab seems to suggest that the synergy, that you don't actually need loop extrusion, but that condensin enhances the simplification of topoisomerase. I don't have time to tell you that part of the story, but I can show you later if you want. But yeah, so we're still trying to figure out what exactly is. But it seems that there is a little bit more to this story. That it's not only this model. It's not that this simple. Now, the second part of the story is about the kinetoplast DNA, which is a pretty fun system. And here I have to kind of mention the collaboration with Kees Dekker, Luca Tubiana, and Akim Schnauffer in Edinburgh. And Sam has joined recently the lab a few years ago, and he's been really pioneering the AFM, so atomic force microscopy measurements of kinetoplast DNA. So he's got a lot of credit for this part of the work. So what is kinetoplast DNA? So this is the mitochondrial DNA of a single cell blood dwelling parasite. So this image here, these are trypanosomes. So these are single cell parasites. They cause a number of diseases like sleeping sickness, I think it's called. Yeah, and Chagas disease as well. So they're quite nasty, and here they're pictured in blood. So these are blood cells. So they reside in blood, and then they get basically spread through mosquitoes. And at some point, people were interested in basically eradicating them, and they started to look at their DNA. And what people have looked at the mitochondrion. So the mitochondrion of this parasite. So they've just one, and they extracted so this part of the parasite. They extracted DNA from that. So let me show you another image. This is the same image that I'm showing you here. So there is this DNA dense here region in the TM. And so if you get that part, it's like a disc. It's like a disc, a DNA dense disc. Okay, like a pancake. Sorry. Oh no. Okay. And so people were interested to look at this structure, and don't ask me why, to be fair, because I mean there are so many structures that one could have looked at, but people have started to look at this. So you extract this structure from the parasite, and you spread it on a grid, and this is what people have found. So in particular, Larry Simpson was the first to do that. So you see that let me just, this is a zoom in of, this is Leishmania tarantula. It's a trypanosome. And what you see is that this genome is not really made of long long string of DNA. These are circles of DNA. And if I zoom, let me zoom on this one, right? If I zoom on that one, what you can see is that these rings are also linked. So what people have started to call this structure, if you imagine the whole, these are just two rings, but because they were broken from the network because during the extraction. But if you think about the whole structure, it's a massive structure that people have called an Olympic network, or also a medieval chainmail. So this is the only DNA that's genome that self-assembles into a medieval chainmail that we know of. And there are a lot of people asking what's the evolutionary benefit of doing so, and this is still kind of ongoing research. Now, we want to understand a little bit better the topology of this network, right? They are linked, but how? What's the exact topology of the network, right? So what we did was to essentially, this is something that we kind of developed in our lab. So you run a gel, but when you load the kinetoplast in the gel, because it's so big, it remains actually in the well. So it's a perfect way to wash any other kind of form of impurities. So you run a gel electrophoresis, you load the DNA on the top here, you run it, and then you can recover it from the well. You absorb it on mica, and then you do AFM. And this was really kind of, and these are kind of the images that you get. This is not the best image, so the scale bar here is 5 microns, but is one of the first that we got, and I'm quite fond of this image. we can really zoom in and start to see all the, so these are all, this structure here is made of DNA rings, like these ones here, DNA rings. These are were these are out of the structure because they were broken, but here they're all linked together. And these are obviously great images, but we want to understand a little bit better the topology, and it's difficult to reconstruct the topology from images like this. So what we did was to do some molecular dynamic simulations. So you take, so to do that, you take a DNA circle outside the network, you binarize it, and then you start to do a simulation where you have some, say you convert these pixels as beads, and you know that those beads represent the DNA in the AFM. And then I have a ring here, the orange ring, that is my DNA circle that I'm gonna let it kind of go, do the simulation, being absorbed, being attracted by the underlying AFM beads, which is the ground truth. Okay, and here you can tell that eventually, if you run the simulation and you quench the temperature, at some point this orange ring looks very similar to the one from the AFM, which is great. It's simple. It's not. But this was just a test. What we actually want to do is we want to do it on the complicated system. So we take a patch of kinetoplast DNA, the internal part. We have these blue beads which represent the AFM beads, and now we put a number of rings, which more or less back on the envelope calculation is the number of rings that we expect in that patch, and we let the simulation go. So we increase the temperature and then we do a quench in such a way that the orange rings are attracted by these blue beads, and then at the end we have a number of configurations that are compatible with the underlying AFM. But now I know perfectly the topology, and I resolved the topology because I let the rings displace also in Z. So I know precisely where the rings are, and I've resolved all the crossings as well. And from these kind of simulations, they are compatible with the FM image, but they allow me to do more topological analysis. So from these simulations, what we do, we compute the Gauss linking number. We compute this quantity here. Essentially, you do the integral over two curves, and this object represents how many times one curve is linked to the other curve, if they're linked. Otherwise, it's zero if they're not linked. And that, so we can compute that, which gives us this. Let me jump to this, which is the most important number. This linking number, essentially, if I do it for every ring, gives me the linking balance. So how many rings are linked to any other ring in the network? And what we find is that they are distributed, and distribution is picked at 3. So each ring is linked to another 3 rings. Okay, so the linking balance is 3, which is the same as what Cozzarelli found in 1995, but through a completely different method. And now we have also a distribution of and the network topology as well. Okay, so what do we learn? I think there is one interesting kind of lesson from this, which is, if you think about it, so this is essentially this is a two-dimensional membrane made of linked rings. Now, if you think about with a valence tree, now I wanted to kind of try to model it. And if you think about essentially nodes and edges, this in 2D essentially corresponds to a sub-isostatic network. So if you think about the Maxwell criterion, if you have a lattice with nodes and edges that connect, or kind of springs that connect these nodes, the Maxwell criterion says that if you have the same degrees of freedom than constraints, then you are isostatic. And to do that, to have the isostatic criterion, you have to have 4 springs, essentially 4 constraints per node in 2 dimensions. But here we have 3 constraints per node, which each node is essentially a ring. And then the number of, and each link is an edge. So it's a sub-isostatic, which means that is a floppy, so has zero energy modes. And so we started to do some analogy with other zero energy, so with other sub-isostatic networks, for instance, collagen networks also have that. And through similar calculations, we could estimate the bulk modulus so we wanted to know the material properties of this network. So the Young modulus, we can estimate it as a sort of if you know the density of the rings, which we know, the elasticity of a single ring, which is essentially given by the inverse of its radius of gyration. This is a polymer physics argument, is proportional to kBt divided by the how big it is, and that gives you more or less the spring, the entropic spring. And then there is this factor here is order unity, which is essentially how far you are from the isostatic point. And this gives us more or less 0.1 piconewton micrometers, which is 10 to the minus 7 newton meters. And I'll remember this number because the bending rigidity you can estimate by the Young modulus times the thickness of the network, and this is more or less the thickness of the network is more or less 100 nanometers, a little bit more, but you get something on the order of 10 to the minus 21 joules. And so what we wanted to compare it with the lipid bilayers, which is the typical membrane type of soft matter. People or biophysics people think about lipid bilayers, and there you get a Young modulus that is 1 Newton meter or 0.1 Newton meter, which is a factor of a million. So our kinetoplast DNA is a factor of 10 to the 6 smaller and a factor of 10 to the 3 softer in terms of bending. So this is what we think we have here. Because of this link, because of the links, you don't really have springs, and in lipid bilayers the molecules are really kind of touching. So here you have a lot of space in between the rings. There is a lot of free space where they can move, but they're still linked. Okay, and this allows the network to be ultra soft. Now this is an estimate based on the image itself. We didn't do any stretching yet experiment, but this is what we would like to do next, right? So in this slide, actually, so now Sam comes in, and what he's done is a number of things. So one is to digest the network. So you can, now we know, so we sequenced the genome, so we know which sequences belong to each circle, and we can cut it using restriction enzymes. And by cutting a different portion of the network, we can measure how the different rings contribute to the shape and the structure. So for instance, if we cut it with ECOR1, it cuts a lot of sequences, and the network starts to be all floppy and kind of weird looking, rather than very different from the control. We can also compare it across species. So this is a T. brucei, it's another trypanosome, T. brucei kinetoplast versus a C. fasciculata, Cristidia fasciculata network. So they look very different, and we want to understand what's the different topology. And then here on the top right, so we also started to play with an optical tweezer and try to really stretch this kinetoplast DNA, although it's a bit tricky experiments because it's difficult to exactly grab this kinetoplast in the right way. So typically kind of and you don't actually know precisely where you grab it, like in more traditional optical tweezer experiments with DNA. So that experiment is a little bit tricky, so we don't have much data yet. But one thing that we can do, and I'm going to skip through these slides because otherwise I'll be too late. We can, I'm going to tell you just one thing about these slides. We can put some quantum dots. So by using dCas9, so this is Cas9, essentially it's an enzyme that recognizes DNA sequences. So dCas9 does not cut the sequence, but we can functionalize it with biotin and then a quantum dot that is very fluorescent. And so what we can do is essentially to take images of this kinetoplast DNA and the quantum dots localized at given sequences that we want. And the sequences that we tagged were the maxi circles. And so what we saw is that the maxi circles are some DNA circles that are very long. So this network, this kinetoplast DNA is made of small circles and large circles. These large circles appear to be localized at the periphery. And what we did was, well, let me skip through this just a second. What we did was to track, so we can take movies where we track essentially the green is the whole kinetoplast and the red is the quantum dots that are tagged on the maxi circles. And what we can do, we can track the dynamics of the quantum dots, and therefore we can track the dynamics of the maxi circles within the network. And so for instance, we can measure the position in X and Y, we can measure the distance between pairs of quantum dots, and through that we can measure a number of things. For instance, the mean square displacement, which is how far a quantum dot has displaced by itself, for instance, the blue curve, or with respect to the center of mass, or with respect to the distance between two. Okay, between two quantum dots. And the important thing here that we recently realized is that this G2 is essentially the fluctuations of a quantum dot with respect to the center of mass of the kinetoplast. So imagine you have a vector that joins the center of mass and one quantum dot, and essentially you look at the mean square displacement of that vector that tracks the displacement of the quantum dot with respect to the center of mass of the network. And if you do that calculation, you can, if you look at this red curve, which represents this value that I just told you, it's between 0.1 and 1 micrometer squared, more or less, on a very large, up to 20 seconds. So, and then it will plateau at some point here. So by doing, again, a rough calculation, what you expect is that the Young modulus is related to more or less kBt divided by these fluctuations here. Okay, essentially, if you think about in terms of a gel, so the Young modulus of the gel will be related somehow to the thermal fluctuations divided by how much the internal gel can move. Okay, squared because of dimensionality, and what we get is something in around 0.0101 piconewton micrometers, which is the same of what we found a few slides before just through the AFM image. So now we're quite happy, and this is a kind of a more direct measure of and still it confirms that the network is really soft. It's an ultra soft material, much softer than any membrane material that biophysics typically considers. Okay, so that's the kinetoplast story, and now I have, let's say, 15 minutes to tell you about, or 10 minutes to tell you about the machine learning part. So this problem is let me acknowledge Joseph was an MPhys, very good MPhys student. Filippo, Yair worked on this project, and now Georgie is working on this project as well. And we had a lot of fun. We had a cover of ACS polymers and physics world, so we had a lot of fun with this project in the last couple of years. We did quite well in terms of also getting published. So I think I was quite happy with this. So so the story goes like this. So back in the days when Peter Guthrie Tate, Lord Kelvin and Maxwell were here in Edinburgh, essentially, where Kelvin was in Glasgow. The problem was, so Kelvin came up with this smoke machine to think about knotted ether. So I'm not sure if you're familiar with this. Kelvin thought that atoms were made of knotted ether, and different types of atoms were made of different knots. And so Tate started to make a table of all the possible atoms together with Maxwell as well. And that was how knot theory started, essentially, how tabulation of knots started. Since then, there's an explosion of knots, and I think the latest one is up to 20 crossings knots or something like this. So it was it's impressive number of knots that you can find through computer, essentially. But the question is, how do you know that they're all different? Okay, that's the big question. So we want to find a topological invariant that tells us whatever knot you're showing me, I can compute something that is unique for that knot. Okay, so if I show you this knot at the beginning, let's say, I want to compute something that tells me what kind of knot it is, right? So if I show you this knot, you maybe say, I don't know, it's a very complicated knot, but if I deform it, it's actually the unknot. Okay. So then I would like to have something, a number that gives me the same number here and here, okay? No matter what's the actual geometry or the embedding of the curve. Some people started to do knot polynomials. So with knot polynomials, for instance, the Alexander polynomial, you can do a sort of calculation, and through this calculation, you can tell that two knots are the same. So for instance, in this case, this is a pair-copair, which for a long time they were thought to be different, but actually they are the same, and this was found through computing essentially the Alexander polynomial. But there are some knots that share the same Alexander polynomial. So for instance, this square, granny, and 820, so these are three foils in the same orientation or opposite orientation, so right-handed, left-handed, or right-handed, right-handed. They share the same Alexander polynomial. Even more strikingly, the Conway and Kinoshita Terasaka, these are two mutant knots, they're called. They are 11 crossing knots, and they share the same Alexander polynomial. So they have Alexander polynomial 1, which is the same as the unknot, which is very upsetting for mathematicians. So we want to find something that is better than what there is now. So this is the big challenge in knot theory. And for my from the polymer physics perspective, I also wanted to find something that was related to the geometry of the embedding. So, for instance, there is a result by Ferry-Milnor that says Ferry-Milnor theorem that states that if the total curvature of a knot is less than 4 pi, then a curve must be unknotted. But this is only a weak constraint on the topology of the underlying curve, because it doesn't say. So I can have a very complicated unknot that whose curvature is larger than 4 pi, but is still unknotted. So my question was, are there geometric signatures that uniquely identify a knot topology? And so we started to investigate on this, right? So we've done a lot of simulations. We computed a number of things, for instance, local curvature or local density. So if you compute the local curvature along this, you get something that is quite noisy. If you get local density, you start to see some kind of patterns. So here there are some peaks, maybe they are correlated to where the essential crossings are. Then we started to look at this writhe. This writhe essentially is the same as the linking number, but now this time is computed on the same curve. And instead of the whole curve, we take in segments. So the segments from k minus L to k and from k to k plus L, so the consecutive segments. So this 1D writhe should distinguish between something that is open like this, or it's super coiled like this. It's kind of coiled onto itself. Or the 3D writhe, you take two segments, so they can be very different, very far apart. And you want to see where that segment is very entangled with the rest of the curve, for instance. And we start to see again cleaner patterns here. And so we started to wonder whether there is something that can be extracted from those patterns. We could figure out that they were localizing with the ends of the knot, okay? But we wanted to figure out whether there is also information on what the knot is. And so the question is, can we extract more information from these type of patterns, right? And to extract information from patterns, one of the ways you can do it is to use machine learning and pattern recognition. So the way we train the network is like this. So we create a lot of knots and we run simulations, very long simulations, and every, let's say, 10 million time steps, we take a snapshot. And that is a sort of a conformation that is independent, and we create 100,000 of these independent conformations of these knots. We compute this segment to segment writhe. So essentially it's the integrand of the Gauss linking number, and it's a function of two variables, so x and y, which is the two segments. For each segment x and segment y of the polymer, we have a value that is computed like this. So this t is the tangent at that point, and r is the position of that point. And we can also, so we get matrices essentially with x and y. And then we can integrate over one of the two variables. So integrate over y of this segment to segment, we get this segment to all writhe. So essentially it's the amount of entanglement that this segment x has with respect to all other segments. And that this STA writhe, so I'll just flip it because it's easier to see in the next slide. So on the y-axis I have segments, on the x-axis I have this STA writhe, and I have different patterns for different topologies, as you can see. And now we pass these patterns to our neural network. We have an input layer where essentially the input layer is the number of bits in that knot. We have a few layers, hidden layers, and then we have an output that in this case is 5 class. So we have anything 0, 1 up to 5, 1, 5, 2. And we want to figure out we train the network to recognize the pattern, and then we essentially test it with unseen conformations. Let me skip through this. So the challenge is the following. So we test it with unseen conformations. What we can do differently is that we can compare how the network does in the sense of recognizing XYZ. So if we pass the network the coordinates of the segments, the coordinates of the segments of the knot, what we get is at best something like 80% accuracy with a feed-forward neural network. With a more complex architecture, we get up to 90%, which is good, but it's not perfect. This is a confusion matrix. For those of you who are not familiar, essentially for each unseen conformation, I know what it is because I pass it to the network. So the true label, let's say that I'm passing it a 4-1, but the network predicts a 5-2 sometimes. And so that is how it gets confused. So most of the times it predicts 4-1, but sometimes it goes 5-2. And so this is the predicted label versus true label, how you read these matrices here. And with the XYZ, you get some confusion here. But when you pass the STA right, you get to 99.8%, and the confusion matrix is just brilliant. So key point number one is that irrespective of architecture, and then neural networks trained with segment to all right are much more accurate at predicting knots. Okay, but now we wanted to challenge it. How about knots that share knot polynomials? So in this case, we got a Granny, square knot, 8-1, and sorry, 8-20. So they all have the same Alexander polynomial, and the XYZ, so if we pass XYZ, the network gets a little bit confused sometimes, but it's still pretty good. But STA right, again, is perfect, 99.9% accuracy. This is more tricky. This is Conway and Kinoshita Terasaka, very complicated STA patterns. They get very, very confused if I pass the XYZ because they're essentially identical apart from one flip here. So the network really cannot distinguish them if I pass just coordinates. But if I pass this STA right measure, this pattern, the network can tell perfectly apart. And remember that these share many, many polynomials, meaning that the network is computing something, a topological invariant, that is more powerful than the Alexander polynomial, for instance. Okay, so this is too good to be true. How do we break it? This is what I asked my students. So we started to increase the number of crossings. So 5 crossing, 6 crossing, 7 crossing, 8 crossing, 9 crossing knots. XYZ gets accuracy less than 30%, so pretty crap. STA right, it stays pretty good, so it goes up to 85%, down to 85% with 9 crossing knots, which is a lot of knots. But here we identified at some point it starts to be confused between a 5-1 and a 7-2. And why is that? So we started to look a little bit closer at these patterns, and we saw that these patterns of STA, they're quite similar. So we saw, okay, we thought, well, this STA is not really the whole story. We are missing information, and the information we're missing is that there is more information in the full matrix. If we take this matrix and we use that as an input feature, there is much more information than just the 1D pattern. This is obvious now. It was not obvious when we started the project, but it's obvious now. And so we started to use this matrix as an input feature, and the network gets back to 99.9% or something like this. And then we pushed it to 10 crossing knots, which is 250 different knots. And if you do it with the XYZ, you get down to 20% or something. And all these knots share the same Alexander polynomial. And they are all and they're all perfectly classified by our network, meaning that it is computing something that can tell them apart. And so it's more powerful than the knot polynomials that typically people use most of the times. So I'm gonna spend 2 more minutes to explain what we think it is. So, and this connects to the work by Eleni. So this is essentially this R quantity, this STS, right? Now, what we wanted to figure out is whether it's computing some kind of new, let me say new with the quotes, topological invariant. So before getting to the new part, we want to figure out whether there is an existing invariant that the network is computing. Perhaps it's an order 2 Vasiliev invariant because it's calculated. You can use essentially the STS right to compose essentially this order n Vasiliev invariant. So we started with order 2. With order 2, we computed, so that you have the true value for all these knots, and then we computed it numerically for all the knots that we simulated, and you get a distribution. But then the important thing is that for each knot, you can train the network to predict what will be the order 2 and order 3 Vasiliev invariants. And what you see is that the true versus predicted is pretty good for V2, but for V3 it starts to be pretty bad. So what we think is that maybe order V2, maybe the network can actually learn, but order 3 it cannot, it really cannot. So there is something different. And what it is different, we think, is that the network is looking at. So let me talk through this plot. So this is the XY, and sorry that is so pixelated. It's really from this week. So this is an XY. This is a coordinates of a trefoil knot. The knot is all clustered here. This is clearly unknotted. If you look at the STS matrix, you see some kind of high point of entanglements down here. And if you look at the saliency is a map that tells you where the neural network is looking at to make a good prediction. Okay, and what the saliency map is telling us is that where the neural network is looking at is not at the entanglements. It's looking around the knot, is looking away from the knot rather than at the knot. And so what we're thinking now is that perhaps is trying to figure out, essentially it's almost like learning Redemaster moves. So sort of saying, oh, is there any twist that I cannot remove outside this knot? So this is a little bit speculative at this point, but this is where we're at. Conclusions. Synergy of condensin and topoisomerase is necessary for DNA unknotting. So I told you this initial story about how the DNA gets unknotted. I told you a little bit about kinetoplast DNA, which is my favorite system to play with. It's a really beautiful and kind of quirky system to work with. And then I told you about the machine learning story, and it seems that there is something to be learned on from how neural networks can classify knots. We're still not quite figure out what it is, but we think that there is something to be learned in terms of topological invariance that we can use for knot classification. Okay, that's all. Let me thank people who did the work, my group, and you for your attention.
Thank you very much. That was a very interesting talk. That was great. Any questions from the audience?
Hi. I wanted to ask. So when you have the AFM images, how does it store the over/under data of the rings?
That's a good question. So there is no so it doesn't store the information. It does. So what I do in the molecular dynamic simulations is that I let it equilibrate, and at some point I let the Z. So I let them displace in the Z direction, and it's completely random what it chooses. Okay.
Good. Yeah, I had that question too.
So in some sense, it's exactly like a tracing sort of algorithm, then with a random displacement in Z, which I agree is not perfect, but in some way you have to figure out essentially where the rings are starting and finishing. So where are the rings in that kind of mess, in the mesh, sort of. Which is not trivial. There are many different solutions, right? There are many ways in which you can arrange rings in that mesh such that you get the image. Yeah.
Other questions?
I have a small question for the machine learning part. I'm just curious, like you always show the result. Are you splitting the data into training set and test set, or
Yeah.
it is just? And so everything was on the test set, so all those results.
Yeah. There is a training, validation, and test, and everything I showed in terms of confusion matrix is test.
And so just a follow-up question for that. I just was curious in terms of the input size. So when you use I forgot what is the name you put, the one that works better. So
Yeah.
is the input size the same, or you had like more, like is it larger
So you
input?
need, for the matrix, you need so the segment is 100, so 100 bits, let's say 100 segments, 100 input nodes. But if you want to input the matrix, you have to have 100 by 100. So you essentially you're inputting more information. That's true. So we're not kind of comparing the same for same, but that's yeah. But still, I mean, the XYZ, if you inputting the XYZ coordinates, is still 3 times the length. So it's also more information than the STA in some sense, but it still performs better. So the point is that this STS matrix is is sort of the best feature, and then understand why is the best feature. So why is the best feature to classify the knots? Is it because it's easy to compute something else out of it, like a knot invariant?
If I want to make your life harder, because I
Yeah.
saw some rules, I mean, it's too good, some reason, but it's good. But what I would like to see is that it's always the same method when you generate those knots. So it's kind of, even if you do trend set and test set, it's still the same routine. So in some sense,
Yeah.
the trend set and test set are
Yeah.
very close together.
I agree.
You have a method that generates a knot completely differently, and you do
Yeah.
training on this one, and you do testing on some very different method to generate.
Yeah.
That might be like super challenging. So
I
really
agree.
like separate.
I agree. Like a freely jointed chain, or even, yeah, what we wanted to do was to try to classify proteins structures using the neural network trained on the polymer model. So because some proteins have knots in them, so some proteins are knotted. So we were thinking about taking the let's say the backbone of proteins and try to classify whether they are knotted. So which will be completely different from, this is something that we haven't done yet, but it's a kind of, yeah, you're right. It would be a very nice test to do. Yes.
Cool. Other questions?
Yeah, I had a question if we have enough time. I actually had several questions, but I might choose one in the interest of time. So first of all, really good talk. I really enjoyed it. And for about the last part, I find it very surprising that it works so well. I'm also very shocked that so the question you're asking about the knots, it's a question about the topology, and it's not a question about the embedding, and it's not a question about the metric. Yet you are trying to measure it with this thing that is actually measuring something that is geometric and captures metric. And it's so weird that it works so well, right? Because you're measuring this thing that has more information, but somehow the machine learning bit is just choosing the topology behind it and then using that. That's super surprising. Why is that working? Do you know, like, why is it working?
I don't know. Maybe, I don't know. Maybe we should sit together with Eleni as well and Kasturi, because I think, yeah, this is. I have a hunch that is related to what Eleni is doing in terms of this Jones polynomial on open curves, but I don't quite know how. But I agree with you. I'm also surprised. There is this concept that you can compute Vasiliev invariance of any order using this right measure. And this Vasiliev invariant is really a topological invariant. So if the network is figuring out how to use this STA's matrix information, which is a geometric information, to pull out a knot invariant, but we're still trying to figure out what exactly is the calculation that the neural network is doing. And obviously it's not easy because it's highly nonlinear and all the kind of. So yeah, that's at the stage we're at, and I know it's quite interesting. Yes.
Yeah, very nice. It does feel like that, right? From the story you told, it does feel like, you know, below the surface lies a topological invariant to be discovered. It's also a little bit shocking that it's doing such a great job while topologists have been trying to do this for such a long time, and then maybe it's actually paving, like, showing a way of how to use this machine learning tools to, you know, find other things that are harder to find just
absolutely.
out of.
Hopefully yes. Hopefully yes.
Yeah. But yeah, wonderful work. Thank you for the talk.
Thank you.
I have a question, but is there another question? I don't want to. Okay, so a question that I had maybe in the direction that, well, I had also many questions, but for the interest of time, what Sebastian mentioned about, like, you know, challenging it with something different, you know, maybe some intermediate step if you compare, you know, same knot types but different length, let's say.
Yeah.
Maybe
So
that's.
our networks so the architecture that we're using is not, you have to use the same length, the same size as the input. But for instance, transformer networks, which is this ChatGPT kind of thing, then you can use. But the problem is that transformer networks are even more complicated. We chose a network architecture was quite simple to try to decode it. If we go down the transformer network, so you can input something, we can train with the length 100 and you can test with length 200, 500, 1000. And you can do it, but at the same time, you use those architecture that are even more complicated. So it's difficult to open that black box. So we are trying to go down kind of simpler thing, simpler routes. We're quite convinced that it's not an artifact. So it's really doing so. Then, since it is not an artifact, I want to try to simplify it as much as possible so that I can actually understand it. Almost like doing the calculation on a piece of paper,
Right,
of what the network is doing.
No, this is very interesting. Yeah, and I'd love to talk about it.
Yeah. No, we should, yeah.
Well, any other questions? Okay, well, for the interest of time, I think we will stop here. And thank you very much. That was really interesting. And thank you for allowing us to record also. I know there are at least some students that weren't able to make it, and I'd love to listen to it later.
It's fine. That's fine. Yeah, it would be useful also for me. So if you can send me the recording.
Yes, I will.
Cheers.
Very good. Thank you.
Thank you, Eleni.
Thank you. Bye.
Thank you all for coming. Cheers. Bye-bye.
Bye.